Hi. So, in previous session we have discussed about the RLR discovery of RLR members, particularly RIG-I, and we discussed about the uh, endogenous ligand as well as exogenous ligand, which is mainly derived from virus, different viruses. And this exogenous infection means the viral infection also result to the disturbance of RNA metabolism, and that result to the activation of RLR signaling pathway. So, in this session we will discuss about the RLR signaling pathway, which is I will show you, or for your understanding I will make it extremely simple. However, this pathway is extremely complicated. Not only this pathway, the previous TLR and whatever pathway you study. I will just touch upon the complexity. I will just show you a couple of a slide in order to understand the complexity of this RLR uh, signaling pathway, as well as the complexity of activation of these members RIG-I and MDA5. And downstream there is also a lot of complicated, but I will not show you those complication. Here I will just show you the complication in activation or deactivation of RIG-I and MDA5. The more complicated pathway is basically not needed to you, it is very much needed to the researcher who are working in these field. So, let us begin with this RLR signaling pathway. So, this RLR signaling pathway is basically activated when there is a virus infection. Okay. So, once this virus is infecting the cell, this RNA is transported into the cell. Initially it will be in endosomes or phagolysosomes, and then this RNA will be also reach in the cytoplasm. Okay, over there this RNA species will be sensed by either RIG-I or MDA5. Okay. So, this is a very simple schematic of RLR signaling pathway. Here you can see that the RIG-I and MDA5, which is initially present in inactive form, as I told you in previous session, that the CTD, the this domain, the CTD domain, carboxyl terminal domain, basically this whole molecule is present in a close format. Close format means neither this card will be exposed nor this helicase will be exposed when there is no infection. But once this infection comes, then this there will be a conformational changes in these molecules, and these molecules will open up in order to sense the ligand and also to activate the downstream signaling. Okay. So, this signaling is initiated through this RIG-I and MDA5, and then there will be a recruitment of adapter molecule. For all signaling molecule, there is a one simple scheme. There will be a receptor molecule, there will be an adapter molecule, and there will be some kinase, and then there will be activation of transcription factor that eventually result to the response, okay, or effector response. Okay, this is a common feature of almost all signaling pathway. So, here the adapter molecule is MAVS, and this MAVS is also known as there are various name of MAVS. So, you will understand that at that time when this RLR pathway was dissected out, there are so many researchers all over the world. They are working in order to discover this signaling pathway, and that's why the MAVS the molecule MAVS or the adapter molecule of this RIG-I and MDA5 pathway, this has a forename. I will show you in a short while. Okay. So this adapter molecule is basically localized over, here you can see this is localized on peroxisomes, okay, and it is present on mitochondrial outer membrane, okay, and it is also present in MAM. This is mitochondrial associated endoplasmic reticulum membrane. Some people use this word endoplasmic reticulum or ER, or some people don't use. Okay, so it's a in a very simpler form, the membranes which are associated with the mitochondria, over there this adapter molecule is present. Okay. So since it is located on the mitochondria, people previously they thought that it is playing an important role in apoptosis. Okay. Probably you might be aware that this process of apoptosis uses the mitochondria, okay, or the some molecules which is present in mitochondria. Okay. So of course a lot of extensive research was performed, and then people do say that okay, yeah, this molecule do have some role in apoptosis. Okay. Anyway, we are not here to discuss the apoptosis. Let's go to the signaling pathway. So once this molecule, the RIG-I and MDA5 is activated, this activated molecule is recruiting this adapter molecule, and here you will see that the RIG-I and MDA5 they have a card, and this adapter molecule also have a card. Okay, and this card is basically interact with the RIG-I and MDA5 card, RIG-I or MDA5 card, and this is basically a homotypic interaction. Okay, and once this interact, then this signalosome, the complex protein, activate both NF kappa B as well as this will also activate the IRF or interferon regulatory factors. Okay, the interferon regulatory factor is IRF3 or IRF7. Okay, as you can see, and the activation of this NF kappa B is this signalosome basically activate towards, and it will activate the IKK complex, as you have seen in previous session in TLR signaling. This IKK complex is consist of three members. Okay, IKK alpha, beta, and gamma. Okay, and then once this complex get activated, this will phosphorylate I kappa B, and this I kappa B will undergo proteasomal mediated degradation after ubiquitination, and free NF kappa B will be translocated into the nucleus, and then there will be a transcription of inflammatory cytokine. Okay. On another hand, this if you remember the TRIF mediated signaling, so TRIF activates the kinase TBK1 or IKKI or IKK epsilon. Okay. So once this kinase get activated, this kinase will phosphorylate interferon regulatory factor 3 or 7. Okay, and then once it is phosphorylated, then this IRF will make a dimer and translocate it into the nucleus, and then there will be a transcription of type 1 interferons. Okay, so in that way this signaling pathway activates. So this adapter molecule, which here you can see the MAVS, they have again they have this card domain. Okay, the card, this has a card as well as transmembrane domain. That's why this is localized on peroxisome, mitochondria, or outer membrane of mitochondria, or the membranes associated with mitochondria. So this adapter got several names because of discovery. This adapter was discovered by 4 independent groups in the world. okay, and they gave their own name. So that's why this molecule has a 4 name, okay. One is MAVS, this is mitochondrial antiviral signaling protein, okay. Another is VISA, this is virus induced signaling adapter, okay. And another name is this CARDIF, which is stand for card adapter inducing interferon beta, okay. And there is a molecule, the another name is finally the other name is IPS1, this is interferon beta promoter stimulator protein 1, okay. So this molecule got several name, and we also discovered this molecule when I was doing research in Japan. So we discovered this molecule IPS1, this molecule, this adapter molecule, and we gave this name IPS1, okay. So this is the very simple presentation of RLR signaling pathway. Here just I will give you the glimpse of complexity. So here you can see this how complex this signaling pathway is. There is a RIG-I, okay, and this RIG-I get oligomerize upon activation, RIG-I or MDA5, and then there will be a recruitment of this adapter molecule known as IPS1, and this is regulated by plethora of molecules. There are so many molecules, and these molecules are protein, and these protein as a regulator, and there are some long non-coding RNA which is also regulating this pathway. At the bottom you can see there is a one more molecule which is regulating the transcript of RIG-I, that is there is a one microRNA. If you see carefully at the bottom, then you will find out that there is a molecule known as microRNA 485 which regulate the expression of RIG-I. So we have discovered this molecule, this microRNA 485, and this molecule plays a very important role in regulating the RIG-I as well as this molecule, this microRNA 485. Very interestingly, we found out that this also control the replication of influenza virus. And how it is controlling? Basically, they directly binding to the transcript which is essential for making polymerase, okay. And this polymerase is needed in order to make the copies of influenza virus. So this microRNA bind with those transcript, and since it is binding, so there will be a reduction in expression of those transcript, and in that way the viral replication is very strongly checked by this microRNA 485. So this is a one snapshot of signaling, and if you see this RIG-I and MDA5, these molecules, these both molecules are very tightly regulated, as you can see in this slide, okay. There are so many post translational modification which activates the RIG-I. There are so many post translational modification which inactivate this these members, that is RIG-I and MDA5. So overall, what I want to say that this is the extremely complex signaling pathway, and in your current scenario, you need not to worry about that complex signaling pathway. And if you are interested and if you want to pursue research in this field, then of course you need to understand these complexity. With this, I am stopping here, and in next session I will discuss about the RLR and disease, and I will also discuss about therapeutics. Thank you.